In this slide, we will talk about transport proteins, and later we will start talking in more detail about each one of these ones. But I want to give you a big picture, and I want to tell you that we will see in the membrane a lot of proteins that have different functions that they can use as a tunnel. Some of them will require they will require energy. So for example, if you're moving ions, some of them will be using facilitated diffusion, and they will speed up the process of facilitating diffusion. So while you can, for example, transport water through the membrane, sometimes you need more water. So you can have special channels called aquaporins that can help the water to cross from one side of the membrane to the other side of the membrane. So to summarize, the first thing that I wanted that you remember is that there's a wide variety of transport proteins, and there are two types: channels, that we will talk in a second, and also carrier proteins. And they allow the passage of hydrophilic substance across the membrane. As I said, some of them they can carry certain molecules or ions, and, and they can use these structures, these proteins, as a tunnel. And in particular, I wanted you, so this is the second point that I remember, that they can transport ions. And the third part that I wanted you to remember is that there are some specific channel proteins that were found a few decades ago that are called aquaporins, that they facilitate the passage of water. So we're going to talk about transport proteins. They're doing a wide variety of functions and moving in a very specific way certain molecules.
Now we're going to talk about facilitated diffusion, that is a type of transport that is still passive, but is helped by proteins. So in this case, in a facilitated diffusion, the proteins are speeding up the passive movement across the membrane. So this is just as a corridor, as a channel to allow that molecules can cross, but it still doesn't require energy. So this is what is considered passive, and in your textbook you will find very similar schematic. These molecules will move through this corridor using this channel protein, and as I said before, there will be corridors that allow specific molecules or ions to cross across the membrane. Examples of channel proteins include the aquaporins, that possibly are one of the most important ones because they facilitate the diffusion of water. As we said before, water can cross through the membrane. However, if we need much faster the diffusion of water, we can use these aquaporins that they go from one side of the membrane to the other side. There is also example of ion channels that they can open and close in response of a stimulus. So they are gated channels. And also we can have some carrier proteins that, opposite to the aquaporins, they cannot extend through the entire membrane. So this can be an example. If you see in this animation, the carrier is suffering some conformational changes in the shape, and is moving from one side to the other side of the membrane. In your textbook, it will look like this. And remember, carrier proteins undergo a subtle change in shape, well, in other words, in their conformation, that translocate the solute binding site across the membrane. So remember, I gave you an example of channel proteins that are important, that they were the aquaporins. Then another example was the ion channels, and finally this third one, that is a carrier protein that it needs to suffer conformational changes that are very subtle, that allow to go from one side to the other side of the membrane. We're going to talk about active transport and how we use energy to move solute against the gradient. So here we don't have more diffusion. We need to use energy to go against the gradients. In this slide, I want that you use this diagram to try to understand what is active transport and why they need to use energy. And to do that, I want that you observe the amount of sodium and potassium in the extracellular fluid, and also the amount of potassium and sodium in the cytoplasm. So stop the video and try to count the molecules. Okay, maybe you notice that in the extracellular fluid, there is a lot of sodium. If you see a lot of these yellow circles, but very little potassium, and in the cytoplasm is the opposite. And what generally happens here is that through diffusion, we will have an interchange of molecules and they will be in equilibrium. However, in the case of living organisms, we need to get rid of sodium and we have to bring more potassium here. So we have to go against the gradient. And to do that, we need to have some transport proteins that they use a mechanism to go against the gradient, and to do that they require to spend energy, and usually in form of ATP.
Now I'm going to talk about the sodium-potassium pump and how the cell is able to regulate these two important ions. So let's talk about this schematic that you can find in your textbook. And the first thing that I want to tell you is that you have an extracellular fluid and you have a high concentration of sodium and a low concentration of potassium, and here you have the opposite case. However, you need to get rid of more sodium. So how they do it? Because if you see in your textbook, they have this representation of the sodium as a circular structure that is going to attach perfectly here. This way they show that there is a high affinity for sodium. So although there is very little in the cytoplasm, there will be very little here. The little that is there is going to attach this protein because of the high affinity. And the next step that happens is that you spend ATP. As all these three molecules of sodium are attached to the protein, it will cause phosphorylation. And this will cause the third step, a conformational change. As the protein is phosphorylated, this will have a conformational change. And if you see the circles, the perfect circle of the beginning, now they are not going to be as perfect. And what your textbook is showing you, that with this is showing you a reduction of affinity. So now the sodium is not going to attach very easily in this protein. So what is going to be is going to be released outside. So although there is a lot of sodium here in the extracellular fluid, this sodium is not going to attach to the protein, so you don't have a way to keep it in that section. So it's going to go and diffuse in the extracellular membrane and look for a different place far from the protein. Now we are going to have to find a way to do the opposite, to bring down the sodium. And how we do that? Two things that happen here. If you see, as we talked before, here you have again the circles, the perfect circles. So you change the affinity, but you not only change affinity for sodium, you also change affinity for the potassium. So now you have a high affinity. These also change, and this is why the potassium is going inside of the protein and is going to attach. This trigger another change in the shape of the protein. So if you see here, here you got again the circular structure. So the phosphorylation, it produces a conformational change. So you spend energy, and now this is going to attract again sodium. This conformation is going to attract sodium, and they also lost the conformational change for the potassium. So this potassium is going to be released, and again by diffusion is going to move away from this protein, while the sodium is going to go back inside. And this can happen over and over again. Sodium. So let's quickly recap what we just talked. Maybe if you want, you can stop the video and think for a minute. Write the steps. What were the steps that you just talked? Okay, so first step, high affinity of sodium in the protein and low affinity of potassium. So the sodium is going to attach. Then this trigger ATP consumption, and this lead to a conformational change, and now you have low affinity for sodium, and this will lead that there is a release of sodium, but now you have a high affinity for potassium. Here and here, the potassium is going to attach, and what is going to happen is that it's going to create another conformational change. So now you have high affinity for sodium and low affinity for potassium. So they are going to release the potassium, and the sodium is going to attach in these spots again. So hopefully, if you have any doubt, just let me know. But hopefully you understand all this mechanism. If not, you can go out to the textbook, and I will also send a couple videos that hopefully make this more clear. Now I'm going to show you a video that is suggested in your textbook, and this video is going to show in a much more detail what is happening. You don't have to show this for the exam. In the previous one you saw just schematics. Here is how scientists think that it is actually happening, how the proteins are folding, how the different molecules moves, and it's a pretty nice work. Okay, so hopefully you enjoy it, and let's start.
Watch an amazing video showing how the Na-positive, K-positive ATPase undergoes specific conformational changes as it pumps Na-positive out of the cell and K-positive into the cell. The cytoplasmic side of the cell is shown at the top of the video. Notice how 3 Na-positive ions are pumped out of the cell for every 2 K-positive ions pumped in. Credit: Mark Hilge and Stefan Hilge, Netherlands Cancer Institute, Netherlands. Hilge M., Seigel G., Voister G.W., Guntert P., Glor S.M., and Abrams J.P. ATP-induced conformational changes of the nucleotide binding domain of NAK-ATPase. Nature Structural Biology 10, 468, 474.
Oxygen or CO2 can cross the membrane, but sometimes you need to speed up those reactions, and you have special proteins that they can work as a tunnel, yeah, some channels that they can bring, for example, water like aquaporins, or sometimes you need special carriers that they can move some solutes from one side to the other one to speed up the reaction. However, in those cases, as we said, this is still diffusion. There is no need to spend energy. However, in the active transport, here, if you see, we were moving from a high concentration to a low concentration. Here we're going to go opposite to the gradient, so we will use ATP to produce conformational changes, to produce changes in the affinity of the protein to be able to move some ions from one side to another side. Or in the cell biology class, you will see other type of ion pumps. To finish, we are now going to talk about how to transport big